In part 1, episode 1 of Mendelian inheritance, we're essentially going to be covering key vocabulary and key concepts you should probably know when you're starting off genetics. And here I have a couple pictures that, kind of represent Mendelian inheritance. I have chromosome, DNA, karyotype. Well, karyotype's more of cytogenetics, and dihybrid cross. And let's just get started. So, in case you don't know, Mendelian inheritance is just inheritance that follows Mendel's three laws. This is not true for all organisms, but this was named after Gregor Mendel. So Mendel's three laws is really a foundation of this, but we'll get into Mendel's three laws as we progress the video. So let's get into the first slide. So types of fertilization. So let me just read the slide. In some experiments, Gregor Mendel wanted to carry self-fertilization on his pea plants, which means that the pollen and grain are derived from the same plant. So the name is pretty suggestive, self-fertilization as meaning from the same plant. And so, as you can imagine, since the pollen and grain are derived from the same plant, they pretty much provide no variation of the they pretty much provide no variation. But in other experiments, however, Mendel wanted to provide variation to his plants, so he cross-fertilized them or made the pollen and grain come from a different plant. So once again, the name is pretty suggestive. And so he cross-fertilized them by removing the anthers or the parts of stamen that produce and contain pollen in the plant. And he removed these anthers before they could produce any more pollen. So this prevented them from self-fertilizing. And so cross-fertilization produces variation and self-fertilization doesn't. And the names are pretty suggestive. All you need to know is that the difference of where the pollen and grain come from. So dominant versus recessive alleles. No, dominant versus recessive and alleles versus gene. So a dominant trait is expressed in the phenotype. So while a recessive trait is masked by the dominant trait or not expressed in the phenotype, a phenotype is essentially the observable traits of an organism. So the dominant trait is just essentially expressed and the recessive trait isn't. And then a gene is a unit of heredity that encodes for a specific trait. So maybe your eye color, I don't know. And then an allele is a variation of a gene. So let's say we're working with eye color. Your mother might have blue eyes and your father might have green eyes. They're essentially just variations of a gene. And so heterozygous versus homozygous. Heterozygotes contain two different alleles for each trait. So essentially, one dominant and one recessive. They're also known as hybrid, and homozygotes contain the same allele for each trait, also known as true breed or purebred. So homozygotes can be homozygous recessive or homozygous dominant. I have a picture there so you can see. And then a variety that contains the same traits over many generations because of self-fertilization is called a true breeding line or strain. So essentially they're just generations with no variation and they contain the same traits from self-fertilization. Genotype versus phenotype. I think I already explained phenotype, but I'm going to explain genotype now. So genotype is a genetic makeup of an organism, whereas phenotype is the observable traits of an organism. And as you can see, we have homozygous dominant, and then that's the genotype, but the phenotype is actually what's being expressed, and then we have that for the rest of the other pictures. So the Punnett Square. The Punnett Square is a method originally proposed by Reginald Punnett. And to construct a Punnett Square, you must know the genotypes of the parents. You can't construct with just the phenotype. With this info, you're allowed to predict the genotypes and phenotypes of the offspring. And we can do videos of me actually constructing one and doing questions using Punnett Squares. And so here we have one hybrid or one heterozygote, and we have one homozygous recessive, and they're just simply being crossed. And they have different alleles, and yeah. The Mendelian ratios between two heterozygotes. So essentially, these are Mendelian ratios that you probably need to know. So memorizing it is actually not really that hard. So two hybrids will make a 3-to-1 phenotypic ratio. So 3 dominant to 1 recessive, and then They will also make a 1-to-2 ratio. Genotype. I meant to put genotypic ratio, so they'll also make a 1-to-2-to-1 genotypic ratio. So one homozygous dominant, two heterozygotes, and one homozygous recessive, and then this is in a monohybrid cross. In a dihybrid cross, two hybrids will make a 9-to-3-to-3-to-1 phenotypic ratio. So essentially, I'll use an example. When Mendel was breaking in plants, this would be 9 yellow seeds, and then 3 green seeds, 3 wrinkled seeds, and 1 wrinkled green seed. So that's a common example for that Mendelian ratio. And then the genotypic ratio is I don't know why I did not put genotypic ratio, but the genotypic ratio for the dihybrid cross is 1-to-2-to-4-to-2-to-1-to-2-to-1 and 2-to-1. It's actually really not hard to memorize because the 1-to-2 repeats 3 times in that specific order, and so this represents so I you can so if you want to actually see the genotypes, you can probably search them online and you'll probably see them, but I can read off the genotypes. So the first one represents let's say we were using let's use a Mendelian example. So let's say we were working with pea plants. Essentially the one represents our homozygous dominant plus homozygous dominant plus let's actually just forget about that because that's a little complicated, and I should have just put that down, but it doesn't matter. Let's move on. Sorry. So Mendel's 3 laws. There's probably 3 laws you should probably know. The first law is law of segregation, and it states that 2 copies of a gene received by each parent separate from each other during the transmission between parents to offspring. Thus, one gamete may only hold 1 copy of each gene. So this is very important in cellular division and also in other processes that involve Mendelian inheritance. Law of independent assortment states that one gene does not influence the other, meaning that there are multiple allelic combinations that are based off of randomness, meaning that when genes are being crossed, they're assorted randomly. There's no certain answer. There's just multiple combinations. And law of dominance states that for 2 alleles, the dominant allele is always expressed because the recessive allele is masked over the dominant allele, therefore does not show expression. And so essentially the key concept we explained last time, that the dominant allele is always expressed over the recessive allele. And in some cases, and this is a Mendelian inheritance. This is not necessarily true in other cases, and we'll be talking about non-Mendelian inheritance, where we can talk about stuff like epistasis, where this is abides Mendel's 3 laws. So I hope you liked this video, and stay tuned for episode 2.